Hi everyone. In this lecture, we're going to talk about some material from Chapter 7. In this chapter, we look at audition, the body senses, and chemical senses. First, let's take a look at what contributes to audition. Audition basically is a way of saying that we first detect the presence of sound. We are able also to then locate the source of the sound and identify what is making the sound. There are multiple definitions of what constitutes a sound. Sound, first of all, is produced when an object is vibrating, and that vibration moves air molecules. When this happens, we detect a few different things, the first of which is pitch. This is what we call subjective. It is an experience of the frequency of the vibration of the air molecules. So how quickly are those molecules of air moving? If they seem to be moving fast, we perceive the pitch as being high. If they seem to be moving slower, we perceive the pitch as being low. This is measured in terms of hertz or cycles per second. If you were ever in band, for example, or if you played an instrument at any point in time and you've messed with a tuner, then you have measured pitch. And if you can recall that, if you've ever used a tuner, you might remember seeing something like this. So basically a wave in the sound. And again, if it's waving a lot, that's going to be a higher pitch sound or higher hertz. If it's waving very slow, that's going to be more of a low pitch, low cycles per second. We also obviously perceive how intense a sound is, so how loud it is. We're going to get more high bouncing around in amplitude when we measure sound, if it is a loud sound. If it's a softer sound, we're going to get a slower rise up and down, less peaks and valleys in the wave. And again, this is measuring intensity of sound or amplitude. Specifically, this is measuring how much compression the air molecules are undergoing. So the compressions and then the expansions again of the air molecules. There's going to be more compression and expansion happening if the sound is loud. The last thing we look at is timbre. This is sound complexity. A lot of naturally occurring sounds have more of what we call complexity or timbre. For example, the sound of an oboe can sound quite complex. Basically, you can also think about it as kind of being a richness of sound. Now let's look at how physically we perceive sound. Let's look at first the structure of the ear, then we'll later explore the pathway of audition. So the pathway or the auditory pathway from the ear into the brain. So first, let's take a look at the structure of the ear. The ear is divided into three different portions: outer, middle, and inner ear. So sound obviously is going to enter through the outer ear here. This is where sound is coming in via the pinna, which is this outermost portion here, and then will travel through the ear canal until it gets to the tympanic membrane, which is right here. And if you notice, this kind of looks like the head of a drum. This is also referred to as the eardrum. Then we get into the middle ear. This is this small area here behind the eardrum or the tympanic membrane. It contains little tiny bones called ossicles, the middle ear bones right here. There are three of those. There is the malleus, incus, and the stapes. And again, these are going to be vibrated by your eardrum or that tympanic membrane. So the malleus or the hammer connects to the eardrum and then transmits vibrations via the incus or by way of the incus and the stapes to the innermost portion of the ear, which is the cochlea. The inner ear, again, is the cochlea. This is the snail-looking structure here. So the vibrations here in the middle ear bones then sends signals to the cochlea, which is the inner ear. This is filled with fluid, and sound must transfer through that fluid to finally reach the basilar membrane, which is the kind of the lining of the cochlea, and it has hair cells that line it called cilia. This vibration has to bend the cilia in order for you to perceive the sound. Now here on the bottom right of our slide, you can see what is labeled as the organ of the corti. This is basically the cochlea kind of stretched out a bit, that snail-like structure of the inner ear. It's stretched out a bit, and you can see the outermost portion here that is kind of shown in blue right here. This area here, right inside that, is the basilar membrane. So that's what we're taking a look at here. And in the basilar membrane, there are two types of hair cells or cilia. You have the innermost. These are essential for normal hearing, such that they've actually found that individuals that, like for mice, for example, mice in laboratories, the ones that don't have the inner cilia actually can't hear anything. So we really think that these inner hair cells are essential for normal hearing, whereas the outer cells are more what we call effectors. So these affect the structure or the characteristics of the membrane itself. So kind of how this membrane here will kind of bend and flex. The outer cilia are more responsible for that. And this will in turn affect how the vibrations are carried to the inner cilia. Now, if we look closer at the cochlea, that little snail-like structure of the inner ear, you can see within the spiral here the different areas in which detection of sound happens. And specifically, we're looking at frequency of sound waves, so pitch. How high-pitched, so here, the higher range of hertz, and low-pitched here, 200 to 600 hertz. And where on the cochlea, specifically within the basilar membrane, detection of these sounds happen. So this is called coding. So we place coding in terms of high, medium, and then low frequencies. And again, these are detected by different parts of the basilar membrane because the membrane itself flexes back and forth. Now, if you notice, the innermost portion here is what's detecting the lower frequency waves. And then as it moves out, detection goes higher to eventually, with this outermost portion being the hair cells, the inner hair cells that are more responsive to the higher frequencies. And again, this is going to be determined by action potentials that are happening in the hair cells. So a hair cell detects a signal or picks up a sound, it's going to depolarize and an action potential will occur and it will send that signal to all of the neighboring cells. So if we look at rating of coding, so rate of coding for the lowest frequencies. So this is again these hair cells that are what we call the apical or apical end, which is the innermost right here. So next to the apex in the center here. These are the ones that we think are going to be detecting or are more important in detecting the lower frequency sounds that tend to be the more important sounds. Now, if we look at how cochlear implants work, these are the implants that are commonly used for folks that are hearing impaired. These are the ones that are given to young children and adults with either complete hearing loss or very severe hearing loss. And the goal is usually to help them understand speech. So basically help them interact with other people and be able to interpret speech. The goal of these implants is to restore the hearing loss due to damage in hair cells. So hearing loss is really a result of loss of cilia or hair cells. And that's what these cochlear implants try to counteract. And they do that by stimulating different parts of the basilar membrane inside the cochlea. So again, that basilar membrane is on the inside here, that blue area, like right in here of the cochlea. these cochlear implants are meant to stimulate different areas. So for low frequency sounds, it's going to stimulate the hair cells more on the inside toward the apex, the apical end. And for medium, out here toward the outer edge, and then finally the outermost edge for the higher pitch sounds. These implants have little different electrodes that attach to different portions of the cochlea and will stimulate the different areas of the cochlea accordingly, depending upon the frequency of sound that's being heard. Now that we've looked at the basic structure of the ear, how sound travels from the outer ear to the inner ear, and we've looked at the hair cells within the basilar membrane, let's now look at what happens to sound once it actually comes into the ear, has traveled through the basilar membrane. Now what? So that's just the ear. Now it's got to get to your brain. So now let's take a look at the auditory pathway. So if we look at the cochlea here, this is going to send auditory information to the brain through what is called the cochlear nerve. So you see the cochlea here. This spiral here is containing what we call the ganglion, which is just a bundle of a lot of different nerves, a lot of different nerve cells. Leaving the cochlea here, you see this yellow portion here. This is the cochlear nerve. This is what is going to leave the innermost portion of the ear, the cochlea, and go into the brain. Specifically, this is going to be a bundle of axons of nerves that are bipolar, and they are leaving the area of the cochlea, again, via the cochlear nerve ganglion. And eventually, they are going to synapse with neurons in the medulla, which is right below the pons in the hind portion of your brain, which makes sense. This is the older part of the brain, and it makes sense that a lot of reception of auditory information would happen here. So action potentials in the auditory nerve axons, again, that cochlear nerve here, are going to synapse with neurons in the medulla. And that's going to be the first area of the brain that this information reaches. Now we do have both afferent and efferent neurons here. So the excitatory neurotransmitter for the afferent neurons are glutamate, and efferent, or moving away from the central nervous system. These are more inhibitory. The inhibitory neurotransmitter here is acetylcholine. Now if we look what happens beyond the medulla, because the information or the signals here, the auditory signals, do move beyond the medulla. Here we're looking at different cross sections of the brain. We're looking at horizontal and sagittal slices, but we're looking at the same area of the brain here, just from different vantage points. So from the medulla, and this side view here I think is a good view to take first, again, is right below the pons. From the medulla, these axons will pass through a little fiber bundle into the dorsal midbrain, specifically the inferior colliculus, which is right here. And then eventually neurons here will project out or have axons that project into the an area of the thalamus, the medial geniculate nucleus, which is in the thalamus. And then finally, axons from neurons here project into the auditory complex in your temporal lobe. Now here we are looking at the temporal lobe, which we tend to associate with audition. So again, that's kind of the end place where this auditory information gets to. So this core region consists of three separate regions. So we have the core, the belt, and the parabelt. So the primary auditory cortex is really the core area here shown in yellow. The surrounding area is going to be more the association cortex, which is going to help us more figure out what to do with the auditory information that the core area is receiving. So the primary area is receiving the auditory information. Then we have these other areas that are more association areas that are helping us kind of put together that information and then figure out how to act with it. We have what are called two streams here, kind of similar to what we looked at with visual information. the what and where pathways, we kind of have something similar for audition as well. We have the anterior stream, which is more associated with analysis of complex sounds. So we see this type of direction of activity when we are listening to more complex sounds. Whereas the posterior stream, which is going from this area here more toward the parietal lobe, which makes sense because the parietal lobe tends to be very active when we are having to locate items in space. The posterior stream seems to be more active when we are localizing sound, figuring out where a sound is coming from. And again, that makes sense that activity would be kind of leaving the temporal lobe and like moving more toward the parietal lobe if we're trying to figure out where a sound is coming from. Now that we've kind of taken a very quick look at where sound goes from the ear to the brain, let's take a look at how we actually perceive sound. Once it's in our brain, how do we perceive it? Let's look more at our perception of timbre, which again is more of that complexity of sound. So naturally occurring sounds are rich. They have multiple frequencies. So we tend to say they have a rich timbre. There seem to be a lot of overtones with these natural sounds that add to the complexity and give multiple frequencies to that sound. Now, what this means functionally is that there are different portions of the basilar membrane that are being stimulated at the same time when we are perceiving these rich sounds. And that is what really adds to our perception of a rich timbre. Multiple areas of our membrane are stimulated rather than just one. And this is going to create a really unique pattern of activity. That's why we can distinguish between different sounds. That's why an oboe, like I used earlier, that example, the example of an oboe sounds a lot different than the sound of a tuba because it's going to create a different pattern of activity along your basilar membrane. Now, these dynamic or ever-changing sounds like those from a tuba or an oboe are again represented by a unique pattern of firing activity along the basilar membrane and are really associated with a different pattern of firing as the frequencies within that sound change. If we look at how we figure out where a sound is coming from, we do best when a sound is coming from a direct midline. So there is nothing blocking the sound from us. So the sound can travel directly from its source to us with nothing in the way. It also is likely to reach one ear faster than the other, and then action potentials will happen there first. We may also change the sound or how we hear the sound by tilting our head. You may do this naturally when you're listening to music or trying to hear a sound from far away or trying to determine exactly where a sound is coming from or what a sound may be. You may notice that you naturally will tilt your head. This will change how the sound interacts with your pinna or your outer portion of your ear. It will change how it actually enters that portion of your ear. And depending on how you tilt your head, you may be able to help the sound enter your ear more directly or in a better way to where you can have a better chance of figuring out where the sound is coming from. This will also alter the timbre of the sound or how the sound affects the inner cilia in your basilar membrane, particularly how firing occurs in the cells along the basilar membrane. And this, of course, is going to differ across people. As we know, some people have damage to their inner cilia, which makes it very difficult, if not impossible, for them to hear. Some folks may have a bit more inner ear cells than others, so inner cilia than others. So this is going to differ somewhat across people. This is going to be really the reason why people interpret sound a bit different. Now we kind of move beyond just basic interpretation of sound to something called the vestibular system. This is really responsible for your balance. I'm sure many of you all have heard of inner ear issues. This is really in reference to the vestibular system. So what is going on in your inner ear and how that correlates with your vision. This kind of makes it possible for you to maintain your balance and not become dizzy and fall. So this is taking into account kind of multiple things. Balance, also the support of your head, eye movements to compensate for your head movements. So as you move your head, your eyes naturally move along with it. That's going to help you not to become nauseous. Now, particularly if we're looking at the vestibular system, we can look at what are called vestibular sacs. These are right in this area here, if you look to the right of the screen, right here. These are connecting off of these semicircular canals. These are really going to help respond to gravity and inform your brain about how your head is oriented. So if you've tilted your head a certain way, your brain is going to get signals that you have tilted your head a certain way. And really, all of this is really important, again, to help you not to become nauseous or not to fall down. So low frequency stimulation of these vestibular sacs can cause nausea. And again, the system is in place to help you. So the goal of this system is to maintain kind of business as usual homeostasis. So this type of stimulation would be a signal that you need to do something different because you're feeling nauseous. Unfortunately, if someone's vestibular system isn't working appropriately, this may happen when it's an inappropriate time to happen. If we look at the semicircular canals here, which I highlighted a few seconds ago up here, these are meant to respond more to acceleration, so your head rotation. So if you're moving your head quickly, these are meant to help your brain acclimate to that and to acknowledge that you have moved your head quickly. And stimulation of these canals can produce dizziness and also rhythmic eye movements. So again, this is just a response to natural movement that you're doing. And again, the goal of this system is to help your brain keep up with what your body is doing. Now, if we're looking at the vestibular pathway, if we take a look over here on the right-hand side of the screen, we have those canals that we were just talking about that are kind of attached within the inner ear. And then there's the cochlea, our little snail guy here. Remember, we have the cochlear nerve that we mentioned earlier leaving the cochlea, but then we also have another nerve that's leaving the semicircular canal area. This is called the vestibular nerve. So together, the cochlear nerve and the vestibular nerve come together and constitute the 8th cranial nerve. If you remember the cranial nerves that we talked about earlier in the semester, this is cranial nerve number 8. Now, most of these project again into the medulla, which is right below the pons in the hindbrain. And it makes sense then that this 8th cranial nerve that's composed of the cochlear nerve and the vestibular nerve also connects in with the 3rd, the 4th, and the 6th cranial nerve. These cranial nerves are involved in eye movements. And this makes a lot of sense because your eye movements need to be coordinated with your head movements. And if they're not, that's going to make it very difficult for you to maintain your balance, for you not to feel ill. So it is important that these kind of communicate and interact with one another. Next, we look at what are called somatosenses. This is in reference to both the surface of the body and the inside of the body. So this is going to be cutaneous sensations, so that those sensations that you feel from your skin. These are going to be the most studied of the somatosenses. So we know the most about your skin senses. But this will also include information about your body position, which is called proprioception. This is the ability to detect what position your body is in at any given time. Also, kinesthesia. This is in reference to body movement. So detecting that your body is moving and how it's moving. And then also what we call organic senses. This is going to be more internal senses. This is information in and around and from your internal organs. First, let's focus on cutaneous, those skin senses. This is going to include changes in temperature, pressure, vibration, and pain. We detect pressure on our skin by the deformation of skin. So your skin cells actually can detect when they're being compressed by pressure on your skin. They're being actually compressed down. They can detect that. Pain is most commonly detected by tissue damage, so actual damage happening to your skin. Kinesthetic information is a feedback from skin stretching. So movement information, we get that feedback from the sensation that your skin is actually stretching. So if you stretch your arms out, your skin stretches as well. And that is going to inform your system that you're actually moving and your skin is being stretched. Some organic information, so that information that's coming from your organs, is felt, but some isn't felt at all. For example, stomach aches, you feel that. You feel detection of that. Sometimes you may get more pleasurable feelings, such as if you're very, very cold, for example, and then you drink a hot cup of coffee or a hot cocoa, that could be more pleasurable. And you may get a sense of that, a pleasurable feeling versus a not pleasant one like a stomach ache. But there are some senses that you may not actually feel from your organs at all. Now, if we take a closer look at the skin, your skin does a lot for you. The first thing is thermal regulation. So this is really what is happening when your skin is trying to maintain body temperature. So we may sweat or we may actually restrict circulation depending upon what temperature is being detected by the skin. Sweat is going to help your skin maintain a cooler temperature, whereas restriction of circulation is going to help your skin maintain a warmer temperature. And this is going to be completely external temperature dependent. We do have different sections of our skin called glabrous skin. This is hairless portions of your skin. So this would include things like your palms of your hands and the bottoms of your feet. These are very dense, and there is a mix of different receptor types in this area. We also, of course, have different layers of our skin. We have the innermost, which is the subcutaneous layer. This is going to be a fatty layer here. Then we move out to the dermis, and then the outermost is the epidermis. And within the epidermis, we see a lot of different types of skin receptors. This is a table taken from your text. This is laying out the different types of receptors that you have, where they're located. So if they're in glabrous, hairless skin, or if they're in the portions of your skin that do have hair, and then also what the function of them as a receptor is. So if we look at the first one, Merkel's discs, these exist both in the glabrous areas of your skin and the hairier portions of your skin. And they're meant to detect roughness, particularly from things like fingertips. So I would say just to take a look at these and just explore a bit about the different types of receptors that you have in your skin, where they tend to be located, and what they tend to be responsible for. So focused on the skin receptors, we think we've identified about 6 different thermal receptors in mammals that are meant to detect variations in temperature. There are chemicals that can mimic these temperature changes without them actually happening. One example would be menthol. So if you've ever used something like Carmex lip balm that has menthol in it, you'll notice that it produces a very like a cooling, almost a tingling effect on the lips. It's mimicking a cooler temperature, but it's not really changing the temperature on your skin. The opposite direction would be capsaicin. This is something that's present in hot peppers. So if you've had hot sauce, it's likely you've encountered capsaicin. It mimics an increase in heat and can trip up your receptors without actually increasing the actual temperature of the skin. If you noticed from that table that I showed you a few slides back of the different skin receptors, there were some that were called free nerve endings in the skin. These are mostly pain detections or pain detectors. So we've found at least 3 different types of nociceptors. These are just nicknames for receptors that are meant to detect noxious or kind of stimuli that are unpleasant. These would include those that detect pressure on the skin, also those that detect heat or acid or capsaicin, and then also those that detect pungent irritants. So things like mustard gas, horseradish, tear gas, anything like that can depolarize these cells and cause action potentials. Here we're taking a very quick look at the somatosensory pathway. Now remember, we're talking about peripheral sensory information. So you are talking about skin senses and organ senses. These are from your peripheral nervous system, not your central nervous system. So all of these inputs are going to come in through that dorsal root ganglia down here. If you all remember, in through the dorsal root and the spinal cord. And eventually this information is going to, by way of a lot of different axon bundles projecting into different areas, going to end up in your primary somatosensory cortex here in your brain. So in through the dorsal root of the spinal cord, and then from the endpoint here, the somatosensory cortex, then information will, or signals will leave here, and then eventually through your spinal cord, come out through the ventral root to your peripheral system. So for example, a skin sense on your hand, let's say, that information will enter through your dorsal root, eventually wind up in your primary somatosensory cortex, and then your association cortex for your somatosensory cortex. So your association cortex that's going to kind of put together that information that the primary somatosensory cortex is receiving, figure out what response needs to be made. That signal will be sent from that association area all the way through the ventral root of your spinal cord and then back out to your hand, which will result in your hand maybe withdrawing or moving in some way. The latter portion of this lecture will focus on more of our perception and management of pain. So first of all, let's look at perception of pain. So why do we actually feel pain? Well, it can be just that we need to take constructive feedback. For example, if we're feeling pain from some stimulus in our environment, for example, if we have something that is, let's say jabbing us in the back, we may want to move. It may be good to move away from that painful stimulus. So a lot of times it's just providing constructive feedback that you need to change something so you don't have damage that happens to your body. There are perceptual effects and behavioral effects of pain. So remember, there's a sensory component of pain, which is perception, the actual perception of pain. There's also the emotional consequence of pain, so how much it actually bothers you to be in pain. Then there's also more long-term implications of chronic pain. Here we're going to take a look at areas of the brain that seem to become more active when we are perceiving pain. I want to, in particular, draw your attention to 2 areas here on the right-hand side of the screen. Here we have the anterior cingulate cortex. This is in the frontal lobes. And then right below the lateral sulcus here, we have the anterior cingulate cortex. Here it's labeled as the insula. This is in the temporal lobes. So what we have found is that painful stimuli seem to activate the insular cortex and the anterior cingulate cortex. So both of these areas seem to become more active when we are perceiving pain. In particular, the insular cortex seems to be more responsive to burning and stinging. Damage to this particular area correlates with a decrease in emotional response to pain, which is very interesting. Damage to this particular area seems to be correlated to, again, a lack of emotional response to pain, but not a lack of pain detection. So the individual would perceive the pain, but they wouldn't be as emotionally bothered by it. And in particular, these folks tend not to recognize that it's harmful, so they don't withdraw from the painful stimulus, which, as you can imagine, can result in a lot of bodily injury. The anterior cingulate cortex in the frontal lobes plays a role in a more immediate emotional effects with regard to pain perception. So less activity in this particular area has been correlated with less pain perception. Now, an important thing I wanted to insert here a note, decreases in the perception of pain intensity correlates with the decrease in activation of the somatosensory cortex, which is at the top portion of the brain here, right above the parietal cortex, right there. However, decrease in perceived unpleasantness of pain, so not the actual perception of pain, but how unpleasant you perceive the pain to be, correlates with activity in the ACC, the anterior cingulate cortex in the frontal lobe area here. So direct perception of pain, more in the somatosensory cortex, but how you perceive that pain, particularly the emotional response to it or how unpleasant you find it, seems to be more related to the area here, the ACC and the frontal lobes. Now let's talk about pain perception and empathy. So empathy is basically being able to view someone else that's in pain and kind of acknowledge that they are in pain, and you almost kind of feel that pain yourself. They have found that the same areas that are activated when you are in pain are also activated when you view someone else that is in pain, with the exception of the somatosensory cortex, which again is that area here. So these other areas seem to be active in a very similar way as they would be if you were undergoing painful stimulation, but the somatosensory cortex is not active when you're reviewing someone else who is experiencing pain. And again, those particular areas that are active include the ACC and the insular cortex, like we've been discussing, and then also the thalamus as well. Now let's discuss some long-term emotional consequences to chronic pain. If we focus on the prefrontal cortex area of the brain, we found that damage to this particular area may result in more of an inability of people to be able to see long-term consequences of pain. So they don't appreciate the long-term implications of experiencing pain. Phantom limb is something that has been a kind of a chronic condition. I'm sure most of you all have heard of phantom limb, but phantom limb basically occurs when someone has had a limb removed or has lost a limb and they still feel sensations from that limb, like it is still there. And a lot of times these are painful sensations. So there have been different treatments for phantom limb. So for example, when a limb is removed, nerves a lot of times can't reconnect. So where the nerves are essentially cut, neuromas will form. And one treatment is to cut the severed nerves above the neuromas. Another treatment is to cut the dorsal roots that have been connected to that limb that are meant to bring information into the spinal cord. So those are cut, so no signals will be entering the dorsal roots into the spinal cord. Another treatment has been to lesion the somatosensory pathway in the spinal cord. All of these have been treatments that have been used, and some of them are successful, but a lot of times pain will return. There is a researcher named Mielzak that suggested to look at the parietal cortex in particular to phantom limb. So the parietal cortex is more active in body awareness and body positions. If you remember, the parietal cortex is that kind of where area, where in space, how to move to interact appropriately with other things. It's also quite active in just overall body awareness. And so his suggestion was to use this area and our knowledge of this area to give different feedback to the brain about body position and body awareness. So to give different visual and proprioceptive feedback. One way to do this is to use mirror box therapy. So for example, if someone is sensing pain in an arm or a hand that is no longer there, they can insert the limb that they do have into a mirror box here. And so what they visually see is their hand that they actually have, and also a mirrored version of a hand that looks as though it's the limb that's missing. And what they can then do is interact with this limb with their active limb. And so, of course, they're not actually doing this, but visually, because of the mirror, it looks as though they can interact with the missing limb. So this allows them to, for example, if they feel an itching sensation on this hand, they can try to scratch it with their active hand or their hand that they actually have using the mirror. And that will send visual information that they are actually scratching the missing limb. And this can provide some kind of relief. Now let's discuss analgesia. Analgesia is this feeling of numbness or lack of sensation or relief from pain. We find more analgesia-like responses or analgesia feelings when someone is engaged in fighting behavior, believe it or not, or in copulatory behavior. Your book talks about a rat study in which rats that were in the middle of copulating were exposed to painful stimuli. That actually didn't stop the copulatory behavior, and this has been reproduced in humans as well. And the suggestion here is that the copulatory behavior produces an analgesic-like effect, so pain isn't felt as much. And this could be viewed as just being an evolutionarily adaptive response to pain to encourage copulatory behavior. Basically, what we think is happening in the brain when we are feeling analgesic-like effects is a few things. First of all, there's an increase activity in what is called the paraqueductal gray matter, which you can see this little tiny blue dot here, this gray matter dot here. And there's a decrease in activity in the thalamus to painful stimulation. And what I'm showing you here is just how opioids can provide pain relief. So basically what happens here is you have a typical path of pain throughout the brain, represented by the yellow arrows. And the blue dots here are the areas where opioids can inhibit pain signals. So this would be drug-like effects. So this is just, again, kind of a demonstration of how an opioid can also produce an analgesic-like effect or kind of block perception of pain. As we know in psychology, we love our placebo groups. We can also find a reduction in response to pain by using a placebo. So what they did here, this is a study that was mentioned in your textbook. They had two separate groups. They had one group that was given what they were told to be a higher priced pain relieving drug. And then the other group was told they were also getting a pain relieving drug, but it was cheaper price. They exposed people in both groups to the same level of shock. What's interesting here is that they were given the same placebo pill. They were exposed to the same shock intensity. The folks that were told they were getting the more higher priced regular price drug reported that the drug was working better. They were experiencing more pain reduction, whereas the folks that were told they were getting the discounted drug or the lower price drug didn't say that they were experiencing as much pain reduction. So it's interesting, the perception of pain or analgesia can also be kind of shown with a placebo effect as well. The very last portion of this chapter focuses on gustation. So this is eating behavior. So sensation of taste. So when we are eating, molecules must be dissolved and stimulate taste receptors on our tongue. We have 6 taste qualities that have been identified, or 6 different types of receptors on our tongue. We have those that detect bitter. We tend to avoid those, as well as sour. And evolutionarily speaking, we look at the items that tend to taste more bitter and sour. They also tend to be poisonous. Not always, but a lot of poisonous items tend to read as more bitter or more sour. So evolutionarily, it makes sense why especially young children would be more averse to bitter and sour things. But we can learn to appreciate bitter and sour, in particular as we get older and they are paired with more pleasant qualities like sweet. We also have sweet taste receptors. Sweet usually signifies as safe. Salt is actually something that is necessary for us to consume, not near as much as most of us do, but it is a necessary thing for us to consume for survival. And we do have receptors that help us detect salty things. We also have something that's been identified called umami. This has been associated with MSG, or monosodium glutamate. This is kind of referred to as a meaty kind of a taste. Particularly, it picks up more on amino acid glutamate, kind of a hearty, more of a meaty flavor. Doesn't have to be a meat item to trigger it, but a lot of times it is. And the last one is fat. We tend to prefer high fat things. This is more of a mouth feel thing. The best way I can describe this, because I'm a coffee lover, is when you get a latte, for example, and you get milk, of course, in your latte. You can taste the difference if you get whole milk in that latte versus skim milk, or whole milk versus something like almond milk. Skim milk and almond milk are low-fat milks, whereas whole milk is higher fat. You can taste the difference in the latte, not so much in the flavor itself, but in the thickness of the drink. They have a different mouthfeel. And it seems as though this may be adaptive for us to be able to sense this higher fat content in something, because fat equals calories. And many generations ago, we really needed to be able to consume fat when we came across it because it would help us store calories and survive longer. So it makes sense why we would also maybe have detection or receptors that detect fat in our diet. So I will note here, though, that taste and flavor aren't the same. Taste is just based upon taste receptors on your tongue. Flavor comes from both gustation, so those taste receptors on your tongue, as well as olfaction, or your sense of smell. So here we're looking at the gustatory pathway. Again, back to our cranial nerves. Our gustatory pathway is correlated with the cranial nerve number 7. So information from this cranial nerve also relays through cranial nerve 9 and 10, and these are corresponding more to movement in your mouth and in your palate and your throat. So again, this makes sense why all of these things kind of work together. So your tongue, your throat, even jaw movements in order to eat. Signals from these cranial nerves are received in your medulla, below your pons, and then axons from neuron bodies in the medulla project out to the thalamus, and then finally are received in the primary gustatory cortex, which is the primary receiving area in your brain for information coming from your tongue, from your mouth when you eat. And then information from that primary cortex goes into the secondary cortex, which again is more of that area for figuring out what to do with that information you're receiving in your primary area. So for fun, let's talk about at the end of this lecture super tasters. Super tasters tend to be more sensitive to certain things than your typical person. For example, super tasters tend to be more sensitive to bitter items. These would include things like Brussels sprouts, for example, or coffee. It could be that they have more of these bitter receptors, so bitter reads as stronger to them than it does your average individual. Some pros to this, they may be less likely to smoke or drink. Smoking and drinking tends to trip bitter receptors, and if you have more of these receptors or you're just more sensitive, you're less likely to engage in smoking and drinking alcohol. You may also weigh less. Cons, though, picky. These are your picky eaters. One of my 3 kids, I'm convinced, may be one of these. Picky eater. You could be at higher risk for things like colon cancer if you're a super taster, particularly because you tend to avoid cruciferous vegetables if you're a super taster, because a lot of times they read as being bitter. This would include, again, things like Brussels sprouts, but also broccoli, cauliflower. These are things that are cruciferous that tend to be really good for intestinal and colon health. But if you're not consuming those items because they read as very bitter to you, you could put yourself potentially at higher risk for something like colon cancer because you're not passing these cruciferous vegetables through your system, and it could put you at higher risk to develop things like polyps. You could also be at higher risk for heart disease because super tasters can have a tendency to consume more salt. Now, last bit here, let's talk about olfaction. As I mentioned, flavor is a combination of taste and smell, or olfaction. So olfaction or smell helps us to identify what is food, and also helps us identify what is not food. So what is something edible, what is something not edible, also helps us avoid bad food or inedible food. So things that have gone off, for example, if you have milk in your fridge and you go to smell it, it smells off. Well, that's olfaction helping you out, helping you avoid something that's inedible. In the past, and in also non-human animals, it helps us to identify and track prey. Also helps us to identify folks that are in our inner group, so family and friends. This would be more hormones, pheromones, but helps us identify people that are familiar and likely safe to be around. It can also help us identify a mate. Now, interestingly, we have about 6 million receptors in 2 patches of mucous membrane in our sinus cavity, which is called the olfactory epithelium. So 6 million receptors in these 2 tiny patches that help us detect all of these different scents. Less than 10% of the air actually reaches these paths. We can help increase this by, silly enough, actually sniffing harder. But still a very small percentage of the air actually reaches these patches. Some of these patches, some areas of these patches contain those free nerve ending receptors. And these are really thought to help us mediate bad sensations by irritants that are very irritating to our sinuses. Now, if we look at how we process olfactory information, each receptor cell sends an axon that projects into the olfactory bulb. We can see that here on the bottom right. Some terminate in these regions of the ipsilateral forebrain. So the same side that is being sent, so ipsilateral is the same side. So the same side of the forebrain. So some stop there. But others actually cross to the contralateral side and terminate on the opposite side of the olfactory bulb. And some also travel through different olfactory tracts. This would include going from the amygdala to the hypothalamus, or going through the piriform cortex to the hypothalamus and the orbital frontal cortex, for example. So some are stopping very quickly, some are crossing into the opposite section of the olfactory bulb, and then some are actually traveling all over the brain. And this will be dependent somewhat on what the scent is and how our system needs to react to it. For the last portion of the lecture, let's talk about olfactory receptors. We've identified in humans about 400 olfactory receptors. Could very well be more. we can distinguish around 10,000 different odors. Now, all of these are represented differently depending upon how scent molecules bind to and result in firing of the receptors. So different scents will have different patterns of firing in the receptor molecules. And that's how those different scents are represented. I wanted to include this last bit before I conclude this lecture because I thought it was very interesting. Something that we discovered more recently is that olfactory deficits, so issues with scent, can actually predate other symptoms of diseases by years. So for example, in Alzheimer's patients, you all may be familiar with hearing the term plaques. When they perform autopsies on folks that have had what they presume to be Alzheimer's. And again, we can only confirm that diagnosis after the person has passed. If we take a look at their brain, we can find what are called amyloid beta plaques that have accumulated in the hippocampus. Now, a lot of times what we have found is that folks that have been diagnosed with Alzheimer's, or at least what we think to be Alzheimer's, have actually had issues with olfaction for years before. So it does seem as though issues with olfactory processes can be an indication of a possible presence of a disease in the coming years, or it can be the first symptom of disease. So that is all for this lecture. As always, make sure to take a look at this chapter carefully, and I will be back with you with our next content folder. Have a great day.